Dear students, hope you are all fine. So today we'll see a topic on microbial biodegradation in the course Environmental Microbiology. Microbial biodegradation is a very vast topic because microbes are involved in the degradation of several compounds, both including natural compounds as well as man-made compounds that are present in the environment. And first we'll see some basics of microbial biodegradation, some important concepts, and then we'll move on to the biodegradation of certain specific compounds. So coming to certain concepts regarding microbial degradation, there's a term called recalcitrant compounds or recalcitrants, which means difficult to degrade. So the compounds which are not degraded in nature after it is being released to the environment, even if the environmental conditions are adequate for microbial growth as well as the action of the microbes, these compounds are said to be recalcitrant. That means it is difficult to degrade. So this recalcitrant compound includes both natural compounds and as well as man-made compounds. Some of the recalcitrant natural compounds are, for example, like cellulose, lignin, etc. They are very difficult to degrade. So they are natural recalcitrant compound, although there are certain microorganisms that degrade these compounds. And many of the man-made compounds are also recalcitrant, and the man-made recalcitrant compounds are called xenobiotics. Xeno, we know it means foreign. And the recalcitrant compounds, their peculiarity is that they have high half-lives, which indicate that they are persistent in the environment. So persistence is always measured in terms or expressed in terms of the half-life. That is the time required to reduce the initial concentration of the parent compound to one half of its quantity. So 150 into 10 power 6 tons of recalcitrant organic chemicals are produced every year, and ultimately they find their way into the soil, surface water as well as groundwater. Another term is persistence, bioaccumulative and toxic, that is PBT. This is another concept when we should understand the principles of microbial biodegradation. And the United States Environmental Protection Agency has identified 53 chemicals as persistent, bioaccumulative and toxic. Among these 53 chemicals, 42 are polychlorinated organic compounds and 11 are metals, particularly heavy metals. So the polychlorinated organic compounds also, as they contain chlorine, it is very difficult to degrade. As you know, chlorine has a lot of electronegativity, so it resists degradation. And we have to differentiate between contaminants and pollutants. Usually, pollutants is a term which is referred when these compounds causes health effects. And contaminants are the term when they exceed a particular concentration and they are present and they contaminate the particular environment. And xenobiotics are chemicals synthesized by humans and they don't have any close counterparts with natural compounds. If they have a close counterparts with natural compounds, they will be degraded by natural microorganisms. So as they don't have a natural counterpart, they are very difficult to degrade by microorganisms. A chemical that is foreign to the biosphere, that is not produced by a natural, biological or abiotic source, is commonly called as a xenobiotic. And also all anthropogenic, man-made, synthetic compounds or pollutant, contaminants, recalcitrant, persistent and also toxicants are the other terms used for these compounds. For example, polychlorinated biphenyls, trichloroethylene, polycyclic aromatic hydrocarbons. These are certain man-made synthetic compounds. They are considered as xenobiotics because of their recalcitrance. They resist biodegradation, and also because of the recalcitrance, they persist and they get accumulated in the living systems. And for a contaminant to get biodegraded, it should have certain qualities. Okay. So the contaminant can exist in different forms. For example, in water, the contaminant can exist in 3 different forms, which affects its availability to the organisms. So if a contaminant is present in the environment, for it to get biodegraded, it should be bioavailable. It should be also be in a dissolved form. But if it is in the absorbed form, it is very difficult to degrade. Or if it is in an incorporated form or accumulated in the organism, it will cause further toxicity. That is, we have already studied about bioconcentration and biomagnification. So it is transferred to the food chain. Some other classification of these contaminants are based on the origin. That is, based on the origin, the contaminants can be classified as anthropogenic and natural, and based on the chemical nature, they can be classified as organic and inorganic, and based on the density, they can be classified as non-aqueous phase liquids, less dense non-aqueous phase liquids, and dense non-aqueous phase liquids. And based on the degradation ability, they are classified as degradable, and in degradable they may be simple and complex compounds, and also non-degradable compounds. And based on their hazardous nature, they can be classified as either highly hazardous or moderately hazardous. And based on the health effects, they are classified as carcinogens, teratogens, mutagens, cardiotoxins, immunotoxins, hepatotoxins, and reproductive toxins. And based on the chemical nature, we can classify them as non-halogenated volatile organic compounds, halogenated volatile organic compounds, then non-halogenated semi-volatile organic compounds, halogenated semi-volatile organic compounds, fuels, inorganics, radionuclides, and explosive. So, based on the different chemical structure or their chemical properties, the ability of the microorganisms to degrade also varies. So, how a compound becomes biodegradable and how it becomes non-biodegradable. So, generally, materials or compounds that can pass through the cell membranes are more readily available to the microbes and they are degraded faster. So, how usually microbial biodegradation occurs by means of enzymes. These enzymes may be either extracellular or intracellular enzymes. So extracellular enzymes are secreted outside the cell. So larger molecular weight compounds are degraded by use of extracellular enzymes. And once these compounds are degraded to small molecular weight compounds, they can pass through the cell membranes and they enter into the cell, and further degradation occurs by the use of intracellular enzymes. And also soluble compounds, that is water soluble compounds, are easily degraded than insoluble compounds, but because water soluble compounds can easily be bioavailable. And branching imparts more resistance to biodegradation, because if any particular compound has several branches, then the microbe find it very difficult to degrade. And also microbes prefer non-aromatic compounds than aromatic compounds, and non-aromatic compounds or aliphatic compounds are degraded more easily, because aromatic compounds always have the ring, and initially the energy required to break the ring will be high, so the degradation also will be very slow. So non-aromatic compounds or aliphatic compounds can be easily degraded. And a dispersed compound provide more surface area for attack, and therefore they are degraded better. So for dispersal of these compounds, normally surfactants are added so that they produce more surface area. For example, petroleum hydrocarbons, they are lipophilic, or they are hydrophobic. So to increase the dispersion property, normally surfactants are added so that it increases the surface area and provided more area for attack by the microorganisms. And regarding the bonds, unsaturated bonds are degraded more readily than the saturated compounds. Alcohols, aldehydes, acids, esters, amines, and aminoacids are more preferred than the corresponding alkanes, ketones, dicarboxylic acids, nitrites, amines, and chloroalkanes. So these are the functional groups that are present in many xenobiotic compounds. So microorganisms prefer certain type of functional groups, that is, they are degraded easily. For example, alcohols, aldehydes are degraded more easily than their counterparts like alkenes or ketones or nitrites and amines. And the another important property is the bioavailability. Once the compound is more bioavailable, it will be degraded easier. Even though the compound is there and it is not in the bioavailable form or bio unavailable form, it will not be degraded by the microorganism. So the readiness with which a solute is in the reach of a potential microbe is referred to as the bioavailability. And the bioavailability depends on the nature of the contaminant. The reasons are environmental incompatibility for microbial growth, insufficient nutrients for the degraders, catabolic repression of the degrading enzyme, unavailability of the chemical compounds to the microbes, and other biophysical characters. Next is the biotic environmental fate. What are the different types of pathways the organisms takes in case of the microbial biodegradation? The interaction of a xenobiotic at the site of action in an organism is often a molecular happenstance. That is, the xenobiotic compounds mimic certain compounds which are present in the natural environment. And other concept is the bioaccumulation. Already we saw the storage of compound in the fatty tissue of the organism that results in the accumulation, bioaccumulation, and further transfer through the food chain through the various trophic levels. And biotransformation is also another metabolic process which is also done by bacteria that alters the structure and toxicity of the compound. That is, it will not be completely biodegraded. It will be changed from one form to another. That is called biotransformation. And when we talk of biodegradation, it is a breakdown of the xenobiotic to carbon dioxide and water. We'll see the different mechanisms of biodegradation. That is, biodegradation is a broad term, and within that we can see biomineralization, biotransformation, bioaccumulation, biosorption, and co-metabolism. So what is biodegradation? Biodegradation is a general term where a compound, parent compound is degraded or converted into simpler compounds. And in biomineralization, it is very specific compared to the term biodegradation. The degradation proceeds in such a way that the end products are carbon dioxide and water, that is the mineral counterparts. When a particular compound is completely degraded to carbon dioxide and water, it is called biomineralization. It is complete degradation. And biotransformation is transformation of one type of the compound to another form. So sometimes the form of the compound which is changed will be more toxic or will be less toxic than the parent compound. And we saw about bioaccumulation already. It is accumulated inside the cells. For example, heavy metals. Heavy metals are treated in a way that they are accumulated inside the cells of the microorganisms. And also biosorption. This is also generally applied for heavy metals. Removal of heavy metals, for example, in a wastewater, the microorganisms like fungi or bacteria can interact with the heavy metals and they are sorbed on the surface of the heavy metals. Then another important concept is the co-metabolism. This I'll show you with the help of this diagram. In other biodegradation, usually what happens is the compound that is being biodegraded is utilized as a source of carbon and energy by the microorganism. But in the case of co-metabolism, the compound that is biodegraded is not used as a source of carbon and energy by the microorganism. So this happens coincidentally. That is, the microorganism does not have an intention of degrading a particular compound, but coincidentally this compound will be degraded. Here, that's a classical example of the heterotrophs or methane oxidizing bacteria. Heterotrophs or methane oxidizing bacteria oxidize the methane. That is, it utilizes the methane as its food or its carbon source. So for breakdown of methane, it uses an enzyme called methane monooxygenase. This methane monooxygenase will oxidize the methane and convert it to carbon dioxide. And this will be used by the microorganism as an energy source. However, this methane monooxygenase can also degrade another xenobiotic recalcitrant compound called the trichloroethylene. So the methane monooxygenase, it can degrade methane, and also it can act on TCE and convert it to carbon dioxide. By reductive dechlorination, the chlorine will be removed. So the microorganism does not have an intention to degrade the TCE, but if this TCE is present, it will act on the TCE and degrade that, but it will not gain energy, or it will not get any carbon source by the degradation of TCE. That is called co-metabolism, and this co-metabolism is now being utilized for bioremediation experiments to degrade this recalcitrant compounds in the environment. So when we talk of the how the degradation proceeds, usually this microorganisms are present both in the aerobic condition and anaerobic condition. So in the aerobic condition and anaerobic condition, there is no much difference. The only difference is that oxygen will be present in the aerobic condition, and oxygen will be absent in the anaerobic condition. And in aerobic condition, oxygen will be used as the terminal electron acceptor, and in the anaerobic condition, apart from oxygen, other terminal electron acceptors will be used. So we can see the difference. Aerobic microorganism, it uses the compound as the carbon source and as the nitrogen source, and also essential nutrients, and along with oxygen, it produces new cell biomass, carbon dioxide, water, and the biodegradation will be done in the aerobic condition. But in anaerobic condition, this is all same, except that here oxygen is used as the terminal electron acceptor. Here nitrate or carbon dioxide or iron or sulfate will be used as the terminal electron acceptor in the respiratory pathway. And here, in addition to the new cell biomass, carbon dioxide, water, in the anaerobic condition, methane will also be produced as an end product. So methane is a useful gas which can be used as a biofuel. So that is the difference between anaerobic and aerobic degradation process. So both in anaerobic and aerobic degradation conditions, several compounds are being degraded by the microorganisms. So usually the contaminants serve as a energy source, for example, as a primary substrate. Substrate means as a food source that is available to be the sole energy source, or secondary substrate means it provides energy not available in the high enough concentration, but still it provides as an energy source. And as a co-metabolic substrate, already we saw about co-metabolism, where the compound is not used as an energy source by the microorganism. That is fortuitous transformation of a compound by a microbe relying on some other primary substrate. Already we saw about the example of methane, where methane is being degraded by the methane monooxygenase, and it is used as a substrate, but on the way it also degrades trichloroethylene, but the trichloroethylene is not used as a substrate by the microorganisms. So here, this is the just a pictorial diagram to show how the microbial degradation proceeds. The requirements of the microbial, this, for example, take this as a bacteria, and the requirements of microbial growth are the carbon or energy source, where the toxicants will be used as a carbon or energy source, and the electron acceptor for respiration, it needs a electron acceptor. In the aerobic condition, oxygen will be used as an electron acceptor, and in the absence of oxygen, under anaerobic condition, either nitrate or sulfate or iron will be used as an electron acceptor. Apart from this, it needs other nutrients like nitrogen and phosphorus. Sometimes the contaminant itself will be providing the microorganism the source of nitrogen and phosphorus if it contains, for example, certain pesticides may contain nitrogen and phosphorus, and the microorganism will be using the pesticide itself as a carbon source as well as a nitrogen and a phosphorus source. And also environmental conditions also should be conducive, because in the initial classes we saw about the living conditions of the microorganisms. Certain microorganisms need specific pH, specific temperature, specific salinity, etc. So this condition should be favorable for the microorganism. Only it is favorable, it will take up the biodegradation process. So basically the biodegradation process is a electron exchange process, or even the microbial life itself is an electron exchange process. So it has an electron donor, that is the carbon source. Here in the case of biodegradation, or compound itself, the compound itself will be used as a carbon or energy source, and electron acceptors are for example oxygen or nitrate or sulfate, etc. And here electron transfer occurs, and by the way biodegradation occurs, and here carbon dioxide and water will be produced as the end products. So you see the difference between aerobic and anaerobic. Oxygen is the terminal electron acceptor in aerobic biodegradation. All other biological degradation processes are classified as anaerobic biodegradation, and in most cases bacteria can use only one terminal electron acceptor. But in certain microorganisms groups like the facultative microorganisms, they can use oxygen or also skip to other electron acceptors like the nitrogen. For example, in aerobic, oxidation occurs and co-metabolism, and in anaerobic condition, nitrification, denitrification, manganese reduction, iron reduction, and sulfate reduction, and also methanogenesis. These are the anaerobic pathways of biodegradation processes.